All right, let's talk about the lymph nodes. So the lymph nodes are probably the most important secondary lymphoid organ overall that you're going to get asked about. Now, the lymph nodes are broken up again into three categories. Again, cortex, we can think of that as being the more superficial layer, medulla, the more deep portion of the lymph node. And the paracortex is kind of like the inner cortex, so to speak, of these lymph nodes. Okay, so we'll talk about all three of these. And again, it's important to know, like previously, where are the B cells, where are the T cells? Okay, so again, B cells, we already said, when there's follicles, there's B cells, right? So the B cells are going to be located in the follicles, just like we saw with the spleen. Now, the difference in the lymph nodes is you do have to keep in mind there's primary follicles and secondary follicles. Okay, so this is actually one of those topics that, you know, it seems very detailed, but this is something that comes up quite frequently. So here's a lymph node. The thing that I want to call your attention to in this lymph node, two things. So the first one here, right, you can see that there's this kind of circular structure. There's a difference in the color gradient between, you know, this darker purple and this lighter pink purple color here. This circle here, this is going to be a primary follicle. Okay, so this is going to be a primary follicle. And I'll talk about what that is in just a second. And we also have secondary follicles. Okay, so, you know, this structure here will be a secondary follicle. So what's the difference? So the primary follicle, you can see that it has a pretty dense, dark color primarily around most of it. Okay, so that dense color is going to represent more lymphocytes, right? So this is going to have more lymphocytes that are kind of just sitting around dormant. Dense amount of B cells that are dormant. They haven't really been activated. Okay, that's the concept. That's why they're not moving. They're sleeping, so to speak, if that's an easy way for you to remember it. Now, the secondary follicles, on the other hand, you can think of these follicles as being activated. So I'll talk about how this whole process works in just a second to clear this up a little bit. But the secondary follicles, again, are going to be the ones that are activated. And so when they're activated, think about them moving around. So if you were trying to take a picture of them, right, on this slide, it'd be hard to pin one down, right, because they're moving around so fast. And so you don't get that dense, dormant appearance where you have really darker staining like you see with the primary follicle. Instead, you kind of have this very lighter color in the center of your follicles, and that lighter color is sometimes called the germinal center. Okay, so we can call this the germinal center of the follicle. Okay, and so those B cells that are in the middle that stain lighter, those B cells are undergoing isotype switching and being activated. And remember, what is isotype switching? Isotype switching is essentially where you're taking your standard IgM antibody, okay, which we'll talk about, that's going to be produced initially, and you can change the heavy chain of this antibody to make it IgG, you can make IgE, you can make IgA. Okay, we'll talk about all of this, like I said, in great detail. And you do this by changing the heavy chain. Okay, so the heavy chain is going to allow you to change, you know, the structural integrity, so to speak, of the immunoglobulin. However, what you're going to notice on all of these is that the light chain is the same. So the light chain is essentially what is antigen specific. Like I said, we'll go through this in greater detail, just introducing it here, but that's the concept. So we have activation, antigen switching. There's a lot of things going on in the secondary follicles. They're staining lighter because we can't quite pin down those B cells. They're just moving, they're being activated, and that's just my intuitive way of remembering that. You can imagine, when would you have more activity in these secondary follicles? Well, if you had an infection, right? If you had an infection, B cells are more likely to be activated because there's an active infection, and so you're probably going to see more germinal centers, more secondary follicles. If a patient, for whatever reason, had a disease where they would have a deficiency in maturing these B cells, for example, X-linked A-gamma globulinemia, what's the issue here? Well, you can't make mature B cells. If you can't make mature B cells, right, we might not have follicle formation. So we can see a significant decline in the number of follicles just in general when we have X-linked A-gamma globulinemia, or SCID for that reason. But this classically gets asked about with X-linked A-gamma globulinemia because this primarily just affects B cells, whereas T cells tend to be spared here, and so that's why they like to test that. Again, big point here, follicles, B cells, germinal centers are going to be very classic for activated B cells likely undergoing isotype switching. Classically going to see more of this in the setting of an infection, less of this with diseases that prevent B cell maturation. Now, paracortex, so think about, you know, if you're just looking at this image here, right, so this is kind of the cortex that we talked about, and you can see here would be, you know, a secondary follicle, here would be a primary follicle potentially, and then there's an outer cortex out here, which is kind of what we were just looking at. The inner cortex, though, would be this region closer to the medulla. That's staining a bit lighter here on this image. And that inner cortex is sometimes called the paracortex, okay? And that's where the T cells are going to be located. So paracortex, T cells, follicles are in the cortex, and that's where we'll have the B cells. Okay, so paracortex, we have structures similar to the periarteriolar lymphatic sheaths. We have the high endothelial venules. Okay, and this essentially allows lymphocytes to enter the blood. And let me just kind of walk you through this. So again, you want to think about if you were a cell or bacteria potentially coming into a lymph node, right? You're going to come in through the artery, And then there's going to be some capillaries that aren't really well depicted here. So let's draw them in. So there's some capillaries. Okay, so you might have some capillaries running through this area around the paracortex and the medulla potentially, right? Okay, so let's just blow this up with a cross section. So let's just say that here's my capillary. Okay, so we're just taking one of these capillaries here, we're blowing it up. And, normally these capillaries will have endothelial cells. Okay, so I'm just going to draw some endothelial cells that surround the capillary. In these situations in the lymph node, sometimes we'll have these kind of gaps in here between some of the cells. Okay, so we might have some gaps there, might have some gaps here, might have some gaps here. Let's just say I had a T cell here, right? A couple T cells here. Okay, they're inside of my blood vessel. When they're coming through here, right, they came in through this artery and they want to go into this lymph node. They want to go into the paracortex. Maybe they have an antigen that they want to present to a B cell potentially to allow class switching to occur. Okay, so these T cells are coming in hot, right? Maybe they have an antigen and they need to get here as soon as possible. So what they can do is, instead of them kind of going through, the venous side and eventually getting the lymphatic circulation and getting over to the lymph node, they can just cross over through these capillaries and get into the lymph node, particularly into the paracortex. So that's essentially what's happening here. So these T lymphocytes can kind of jump through, you can think of them as little spaces in between the capillary wall, allowing the T lymphocytes to just get into the lymph node directly. Okay, so that's what they're there for. And these again are called the high endothelial venules. I'll circle it here. HEVs. And remember, those are in the paracortex. Now remember, the T cells, we'll talk about this in great detail, very much associated with viral and fungal infections. So if you had a viral or fungal infection, you would see a pretty significant increase in the density of the paracortex in general. You might see some paracortical hypertrophy. Now, DeGeorge syndrome, remember, we talked about thymic aplasia, third pharyngeal pouch. Remember that. That's associated with DeGeorge syndrome. Now, if you had thymic aplasia, remember, the thymus is where the T cells mature. If you don't have mature T cells, you're going to have paracortical atrophy, right? You're going to have the opposite. Just like if we said, if you don't have mature B cells, you're going to have atrophy of your follicle. So just kind of, this is where you put all of the pieces together by understanding where these cells are from. Okay, so that's the multi-step integration and what we're talking about. Okay, finally, last thing here, the medulla. Again, that's going to be this inner layer. You're going to have your sinuses here. Here you'll see plasma cells, lymphocytes, and macrophages. The idea here is, right, we're bringing in this blood. Eventually, this blood is going to come out through our venous system. It's going to come out through an efferent vessel. And then we also have lymph coming in through afferent vessels and then leaving through efferent vessels. Okay, so we have lymph coming in through lymphatic vessels, and that lymph coming in is going to bring in lymphocytes, antigens, but we also have cells coming in from the arterial side, and that's all essentially getting mixed here. Let's just say that we have a B cell here. Okay, so we're just going to say we have some B cell. All right, so our B cell is going to run in to an antigen. Okay, so maybe we have a bacterial infection, and maybe there's an antigen from that bacteria floating around in the blood, and that antigen is going to run into smack dab into our B cell in the bloodstream somewhere. Okay, so this is all going to be happening in the bloodstream. And let's just say that this antigen has the same epitope as the B cell receptor, and we'll talk about all of that in a minute. But let's just say that that's the case. Essentially, let's just say that these guys are a match. So the B cell, what it will do is it might get activated, potentially. Okay, so this B cell might get activated. Let's put an orange circle around it. This is indicating that our B cell is now activated. So once this B cell is activated, one thing that it can do is it can turn into a plasma cell. Okay, so it can turn into a plasma cell. And plasma cells, you can think of them as factories for antibodies. So it can just pump out a bunch of antibodies against this bacteria, hoping that this antibody runs into that bacteria that had this antigen and then attaches itself to the surface, allowing it to undergo complement-mediated destruction or phagocytosis, like we kind of talked about. So that's one thing. But what the majority of B cells will do is they'll end up going to the lymph node. And since this is an activated B cell, once it gets to the lymph node, it's going to start forming germinal centers. Now, you can imagine, like I said, there's this pale area here. That's going to be the activated B cells. And the activated ones are kind of pushing all the dormant B cells out of the way. And so that's why you get this clear center region, and then you have this dark dormant region around it, because all of the B cells that are not part of the party, essentially, right? They don't have that same antigen receptor. They're going to be on the outskirts, while all of the B cells in the middle, these are all kind of planning their defense from this antigen. Okay, and that's the germinal center that we talked about in the secondary follicles that are activated because they ran into this antigen in the blood. Okay, so that's where the majority of the B cells are going to go, in the lymph node. And that's where this activation comes from. Okay, so that's that secondary germinal center. Now remember what I said, once you get into the germinal center, those cells will undergo isotype switching. So these B cells that are in here, in the germinal center, that are activated, those cells are going to be undergoing isotype switching. So what that means is, now they can make other antibodies. So you might get some of the B cells in that germinal center, maybe some of these B cells will start making IgG antibodies, for example, or IgE antibodies, right? Depending on what this antigen was, right? If it's a parasitic infection, we might need more of our IgE antibodies, for example. So IgE antibodies, for example. So that's all going to depend. Okay, so there are certain cytokines that will stimulate that. We'll talk about it. And this whole isotype switching is going to happen largely because of some interaction with activated T cells. Okay, which we'll talk about, like I said, in greater detail. The idea is that you're also going to have T cells that are going to be floating around here in the bloodstream, right? So T cells can also potentially run into our antigen that's floating around in the bloodstream, right? It might run into that same antigen that can bind to it, and then these T cells might go on into this lymph node to stimulate the B cells to go isotype switching, which would form IgG, IgE, you know, different isotypes here, IgA, etc. Okay, so that's going to happen because the T cell and the B cell will link up, and that will allow isotype switching to occur. And the specific markers that we talk about with this is going to be CD40 and CD40 ligand. Okay, and we'll go into more detail on all of this. But again, just to recap, we had a B cell in the bloodstream, it ran into some antigen, it went to the lymph node, then became very active in the germinal centers. We also had a T cell that ran into that same antigen. That T cell would also come into the lymph node, bind to that B cell. Okay, you have an interaction at a specific receptor, CD40 ligand. That's going to stimulate the isotype switching process to occur so we can turn our IgM antibodies into IgA, IgG, and IgE. Some of these B cells too might stay in the center here and form memory cells. Okay, so then if we ever get infected with this bug again, we would have cells already ready for it. Okay, so some of these B cells would form memory cells, and then some of these B cells here are also going to form the plasma cells that will undergo isotype switching that will create a host of different antibodies. Like I said, we'll go into all of this in much greater detail, but I just want you to have the framework for when we talk about the specific cofactors in the future. Okay, just to wrap this up here, Peyer's patches, just know that they're in the ileum, and know that if you had an antigen out here, that antigen, for it to get into this lymphoid tissue in the Peyer's patches, it would have to traverse this tissue via a specific cell type, and that cell type is the M cells. Okay, so that's important to remember. So the M cells allow the antigen to transport to the macrophages, to the antigen presenting cells, and then these B cells would eventually identify those antigens, right, that are in this lymphoid tissue. And you can see this kind of looks similar to the lymph node, right? You have this area in the center that looks very similar to that germinal center we just talked about. Okay, and so these active B cells would then go on to become plasma cells, plasma cell factories, and produce IgA. Whenever you have mucosal tissue, IgA is the antibody you want to think about. So particularly here in the GI system, so we sometimes call this the mucosal associated lymphoid tissue, the MALT. Okay, so just remember IgA, M cells, ileum, Peyer's patches, and then also just remember that the tonsils are also a secondary lymphoid organ. And these are the references for this video. I'll see you in the next one.